on us. We cannot go it alone, not with mission-based work. You cannot. You have to have a lot of stakeholders at the table. You have to have a lot of conversations all the time with the right people at the right time. And so our sponsors are key stakeholders in this whole equation to getting to better treatments, better quality of life. And so I just really want to thank them on behalf of the organization, our board of directors. Thank you for supporting us in everything that we do. Thank you for showing up all year long. And thank you for all that you do for patients. Thank you. Okay, so what have we been up to? We like to take this opportunity to talk to you about the organization. Like I said, I know we have a lot of new friends in the room. So let me just start by saying MGFA is the leading national patient organization solely focused on myasthenia gravis. We don't have any brick and mortar. We have 15 staff around the country. We all work out of our home offices, and our pillars at MGFA are advocacy, education, and research. As you get to know us better, you'll learn that our activities all throughout the year are really primarily based on education, advocacy. There's so many different ways for you to plug in, whether you attend a monthly webinar or you attend one of our MG walks or a community health fair. This conference, we have an annual scientific session, so a scientific forum that we host. And later this year, we're actually going to host our international conference for the first time outside of the United States. So we are always expanding our footprint, and the organization has grown tremendously in recent years. We've more than doubled in size. And so it's a really exciting time, as we all know, in the MG space, certainly with the movement and the treatment landscape, but particularly for MGFA as an organization. In a lot of ways, we're really growing up. So it's really exciting. And we just love that you're all a part of that growth and evolution. So in the last year, we exceeded the $5 million mark for the first time as an organization. Yeah, it's great. And these are just highlights. So there's a lot of other work that goes on behind the scenes. But as far as education and advocacy, we launched the MGFA helpline, which was part of the initial vision for the organization. And we were so proud to be able to do that. The helpline is used extensively. And we actually have Kevin Mack, who is the CEO of Mack Counseling, which is the provider for our helpline. Kevin Mack, can you stand? Hey, Kevin. Kevin was working with us for the better part of a half a year, and then his teenage son received an MG diagnosis. So talk about paths being meant to cross. Kevin is fully invested, and we're so happy that you're here with us, Kevin. We created international support groups. For the first time, we have support groups outside of the United States. We also, in that same vein, translated MGFA educational materials on our website into 8 different languages. We grew attendance across all of our programs. The National Patient Conference, the scientific session, our community health fairs continue to grow really at an unprecedented rate by hundreds. So like I said, this is just a tremendous time of growth for the organization, and we're making new friends all the time. In regards to advocacy, we did sign on to 9 legislative calls to action, a lot of access to care and disabilities. And so that's something that we want to spend more time focusing on as an organization. And last year, we launched the inaugural stakeholder roundtable. That is where we brought all of our partners in industry together to have a conversation about how we could merge synergies and have more impact together. That was really, I think that was a really special initiative. We're going to continue with that initiative. We have our second roundtable this year. Again, thank you to our partners who plug in and say that it's important to have that discussion together. It's important to come together and talk about how we could be doing more for the community and merging those synergies and resources. And what came out of the stakeholder roundtable, so you know, is a plain language lexicon that we will be distributing to care providers to give to patients so that they can better understand all those terms that you hear in MG that you feel overwhelmed with in plain language. So that lexicon is being developed right now. And then for research, we funded over $1 million in research. And we continue to be on this trajectory. The last 3 years, we've been funding over $1 million in research. This is very, very exciting for us. It's quite a move from just 4 years ago where we really were funding small pilot grants. So it's awesome. And then our MGF grants program expanded globally with our first award ever given outside of the United States, and that was to Dr. Anna Punga out of Sweden. So it was a fantastic year in review. So we just want to share some of those highlights with you guys. Okay, and looking ahead, so what's in store for this year? Well, this year's budget, we're going to hit over $6.5 million in revenue. This is going to be a significant year of growth. We're going to continue to expand our community health fairs. We're growing from 11 to 14, and we're going to have our first international health fair in Canada. If you want to learn more about the community health fairs, please go see Tasha. She's in the back of the room. Tasha can give a little wave. She's your community health fair person. And then we're going to host our second roundtable with our industry partners. We're going to continue the translations of the website and offering our educational materials in other languages because we really need to. There's quite a demand for that from our friends across the borders. We need to be doing that. And then we're going to be developing an MG advocacy toolkit so that people understand. And this is just not just for one time a year when there's like we go to the Capitol and we go to Head to the Hill. This is for all year round to really help people understand how they can be better advocates and have those important conversations. With regards to research, we'll be hosting our international conference, and we're going to continue to expand our research agenda. This is not the actual name for what we're doing, but because it's easier for folks to understand, you're used to this terminology, is we're actually going to be expanding and having centers of excellence, which is a really big deal for the organization and for research in general. So we're really excited about that. Those will launch later this year. And then we're completing what's called the MG consensus guidance, which will become the standard of care that gets in the hands of all providers, MG care providers. So we lead the charge on that as a neutral party to put that consensus guidance together. So that's what's ahead this year. And we'll continue to do all the other work that you know about, like the walks and the webinars, but those are some highlights. Okay. Next slide. So with all that change, for those of you who joined us last night and you saw us give the Ellsworth Award to Brian Gladden, we continue to have some transitions internally, just like I talked about the continued growth of our staff team. We've had some transitions on our board of directors. So I just really want to highlight our new chairman, Robert Thomas, and our vice chair, Justin Stachieris. Robert, if you could just stand, I'd love to acknowledge you as the leader of our board of directors. As I shared last night, Brian's term was up in just a matter of a few weeks, but he really wanted to transition starting last year. So we had been transitioning since about the October, November timeframe. Robert is unbelievable. And what I want to share with you as a community, because I think it's important for you to know, not just to get to know your professional staff team, but also to know about your board of directors. First of all, we have an outstanding board of directors, some of the finest board members I've ever worked with in my career, and I've worked with a number of boards. It's synergistic, it's cohesive, it's supportive, it's compassionate. It's a nice balance of patients and folks who do not live with MG. Robert is just an amazing person, and I'm just like elated that he is our board chairman. And I think he is so good for MGFA and for the community for so many reasons. And so I want to thank Robert for stepping up to take the chairman role earlier than anticipated, and just in general, because it's a huge responsibility to serve as a volunteer in that capacity for an organization. So thank you. At the Yale School of Medicine. But for us, for MGFA, more importantly, Dr. O'Connor is the chair of our Medical and Scientific Advisory Council. He also serves as our chief scientific advisor. Therefore, with those roles, he is a board member as well. What I can tell you about Dr. O'Connor, and you'll hear this and see this yourself, you'll witness this, is that although he runs the MG lab at Yale, and he is one of the most highly regarded scientists in the space in rare disease, he is extremely down to earth. He has this way about him when he delivers his message. You're going to be able to follow him through his talk, And he is very compassionate. He's a very compassionate presenter in person. He attends our community health fairs and our walks. He loves to speak with community members. I think it's a treat that we all have access to him here today. And so I hope you really enjoy his talk. I think that you will for sure. And he's going to talk to us about the current climate and the research landscape. But with that, it is my pleasure to bring Kevin to the stage for his talk.
Thank you, Sam. Another hand for Sam. Okay, let's see about this clicker. There we go. Okay, there is the Slido. So you can get ready to use that if you want. And as Sam said, there'll be microphones afterwards if you don't want to use Slido. You're right, Sam. This is not responding like we want it to. There it goes. Okay, so there's my title slide. And I'd like to start with this. I'd like to start with a little introduction that goes a bit beyond what Sam had said so that we're level set here. Sam, you're right. This is very frustrating. There it goes. Okay. So as an additional introduction, I just wanted to say that I am indeed a professor, and the misconception is that I spend my time teaching. In fact, I don't. I don't spend very much time teaching at all. I run a research laboratory at Yale, and we are investigating how myasthenia gravis works so that we can understand it better. So I spend 90% of my time running a lab, and these are some pictures of the folks in my lab that are all working very hard on myasthenia gravis. So what I'm going to talk about today with all of you is the science of myasthenia gravis. You're going to have some presentations during the conference from clinicians who will talk about that aspect of the disease. But what I wanted to bring to you today was a bit of an understanding about the underlying way the disease works and the different subtypes of the disease and how they're different. And I do hope that it is indeed educational. And if you're seeing something like this for the first time, there's a lot. It may be a bit confusing, but if you really want to understand more about the disease, just keep reading about it. And I hope that this serves as a very good foundation in this Myasthenia 101 that we're going to talk about. And then two short vignettes after that on what's happening in the academic research space right now. And then I'd like to share with you some examples of the research that the MGFA is supporting right now that's meant to help all of us in this room. So let me start with the disease and where it begins. And so we're going to keep referring to this particular space during the next few minutes when I speak. It's the neuromuscular junction. This is the site, this is the place where the disease manifests, where it happens. It's all happening at this neuromuscular junction. So we're going to use it as our home base. And let me give you a few diagrams that can help understand this a bit better. On the right side, you're looking at a diagram of the neuromuscular junction. On the top, there's a nerve, and on the bottom, there's the muscle. And what simply happens is that the nerve sends a signal to the muscle, and the muscle responds. And we're going to go through all the different types of the disease with respect to this neuromuscular junction, this space. And as a mnemonic, or a way to remember, or a way to understand this in a simple way, is that you can consider the nerve to be a letter carrier, a post person. The acetylcholine, which is the molecule that sends the signal from the nerve to the muscle, is like mail. And then the muscle, where the acetylcholine receptor is the mailbox. So it's a simple process: delivery, letter, and then receipt. And that's what's happening in the neuromuscular junction as our nerves are firing and our muscles are moving now. And this will be the home base. We're going to keep coming back to this to talk about the different types of the disease. And so on the right, I'm showing you how does it work. We're going to use the mnemonic again, but the signal, the nerve fires, and it releases this small molecule called acetylcholine. And it travels across the space in between the nerve and the muscle, and then the acetylcholine receptor receives it and the muscle contracts. That's the simple process that's happening in all of us now. But the issue is this: in myasthenia gravis and congenital myasthenic syndromes, CMS, there's an interruption of that process. And we're going to talk about all the different ways that that interruption can occur today. But what's happening essentially is that simple delivery system of the mail person, the envelope, or the mailbox, there's something wrong and the signal doesn't get delivered. And that is the fundamental cause of myasthenia gravis. So how? How is the signal interrupted? Well, there's a number of different ways, and we're going to talk about them in more detail. But at the very basic level, on the left side, you're looking at the neuromuscular junction where the messaging is meant to take place. And on the right, I've showed you two of the major ways in which those signals can be interrupted. The first is the immune system. And so what's happening in many forms of myasthenia is the immune system, our immune systems, which are meant to protect us, are actually attacking that neuromuscular junction and interfering with the signal transduction, the messaging that's supposed to be happening. And that is called autoimmunity. So myasthenia gravis, the AChR type, the MuSK type are autoimmune diseases. We'll talk about those in a bit more detail. The other type of myasthenia that there's folks here, I'm sure, with CMS, is congenital myasthenic syndrome. And again, the situation is that there is an interruption between the nerve and the muscle. And that's happening because of genetic abnormalities. So it's not the immune system. There's a perfectly functional immune system here. But instead, there are individual genetic abnormalities that interfere with either the nerve or the production of acetylcholine or the way that the muscle receives a signal. We'll talk about that in a bit more detail. So those are the two this is a fundamental beginning of where we're going to go. And so many of you know that there are major subtypes of myasthenia gravis. It's a very heterogeneous disease. And when I say heterogeneous and subtypes, what I'm talking about here is the way in which the disease works, or it's called the physiology of the disease. You'll hear from other speakers today, my clinical colleagues, who will talk about thymoma-associated MG, or early onset or late onset, and those are clinical subtypes. We're going to speak a little bit today about the physiological subtypes that are defined by how the neuromuscular junction is interrupted. And this is very important to understand. If anybody's out there and you've been diagnosed with myasthenia and you don't know your antibody subtype, it's important that you try to find that out because the therapeutics that are applied can affect these subtypes quite differently. And what I'll walk you through next is how these subtypes work and how they're different. So let's start with the autoimmune type. So the autoimmune myasthenia includes a number of different subtypes you've heard of: the acetylcholine receptor subtype, the MuSK-specific subtype. There's another one called LRP4, and there's another one called LEMS. It's Lambert-Eaton myasthenic syndrome. Don't worry about the words. They're big scientific words. We're going to break them down and look at how all of these are different so you have a deeper understanding of what's happening at the neuromuscular junction that's causing this disease. So let's start with autoimmune mediated. And again, autoimmune means that you have an immune system that is failing to not attack yourself. So the immune system is meant to protect us from the flu, from COVID, whatever comes along. And then when it turns and makes a mistake and attacks us, that results in autoimmunity. Other examples of autoimmunity are type 1 diabetes, lupus, rheumatoid arthritis, multiple sclerosis, et cetera. These are all situations in which the immune system has gone off track and decided to see us as foreign. And this is what's happening in these autoimmune forms of myasthenia gravis. So a really important concept to understand when we're thinking about all these different autoimmune forms is that they're mediated by antibodies. So this word is essential to understand. So what is an antibody? So this is a protein that's made by our immune system, and the purpose of it is to protect us. And if there was any silver lining to the pandemic is that a lot of folks were exposed to immunology because we heard about antibody titers And we learned a little immunology to understand what's happening. And so I've used on the right side in the example here to show you what an antibody does when it's working to protect us. So on the top right, you're seeing the SARS-CoV-2 virus. And the way that that virus infects us is when it gets in, it binds to a particular receptor in your mucosa, so in your mouth or your lungs. And when you make an immune response to that, your immune system makes antibodies that interrupt the ability of the virus to stick to our own cells and then invade. And so there are these proteins that are really just blockers. They block other molecules from interacting with each other, and they protect us. And when you take the vaccine, you develop antibodies that will protect you before ever seeing the virus. And so in myasthenia gravis, it's the same thing. Here on the left side, back to our home base with the neuromuscular junction, there's our post person and the mail being delivered. The top is the nerve, the bottom is the muscle, and the yellow balls are acetylcholine, the envelopes, mail being delivered. And you can see what I've put in here now on this slide are the red Y-shaped molecules, and those are antibodies. And those antibodies, in this instance, are binding to the acetylcholine receptor. They're not supposed to do that. You're not supposed to make antibodies to self. You're only supposed to make antibodies to things that are foreign to us, which would do damage. And so this is autoimmunity, the fundamental part of autoimmunity. And let's look at a little more detail. Well, how do they do that? Here's a really simple example of how they do it. So antibodies can actually block the acetylcholine from binding to that acetylcholine receptor. So think of it as they block the mail from being put into the mailbox. So on the left side, I've given us our diagram again, but if I draw your attention to the right, you'll see in the left part of the right panel, I show this, the yellow balls are acetylcholine, this delivery system, and they're like a key. And then the blue shaped molecule is the acetylcholine receptor, and that has a lock, and the key fits perfectly into the lock. And that's how the signal is transmitted. But on the right side, I show you this red Y-shaped molecule. Again, that's an antibody. And an antibody can adopt a shape that looks exactly like acetylcholine and stick right into the part of that receptor where the acetylcholine is supposed to be. So the signal's never delivered. The nerve is happily producing acetylcholine, expecting the muscle to move, but the muscle's not there, not receiving the signal because there's an antibody, because of this autoimmune disease, that's blocked the ability for that, for the receptor on the muscle to receive the signal. It's quite straightforward. It's a straightforward system that we understand well. And this is not the case for a lot of autoimmune diseases. We don't understand them this well. And there's a good part of that is because we can target these therapeutically by understanding the disease. So let's take another example. Antibodies do another thing. They activate another part of our immune system that's called complement. It just complements the antibodies. That's easy to remember. And so on this diagram, I'm showing you what happens if you get infected by a microbe, by a bacteria or yeast. You would produce antibodies to protect you from that bacteria or yeast so you don't get sepsis. And it activates another part of your immune system that complements it. And the simple thing to remember here is that when antibodies activate complement, it damages tissue. Not tissue for us, it damages the microbes. It actually kills them. So on the right side, I'm showing you these microbes with these holes in them, and they're leaking out their contents and they die. So that's our immune system functioning to protect us. And the complement system is very powerful and very important to protect us. However, what happens in some patients with myasthenia is the antibodies that we're producing will bind to the acetylcholine receptor. So back to our home base, and looking on the right side, I'm showing you a blow up of the home base. The large blue molecule with the arrow going through it is the acetylcholine receptor on the muscle, and the orange is the orange ball is acetylcholine. And then the antibody binds to the acetylcholine receptor, and it activates complement, this other system that we have, and it damages the tissue. So it's misdirected immunity, it's autoimmunity, and it causes damage to the muscle tissue. So there's another way that the disease can move forward. So let's pause for a minute and leave acetylcholine receptor myasthenia and talk about another form. The other form of autoimmune myasthenia, there's several others, but one of them is MuSK and one of them is LRP4. These are other proteins that are present on the muscle. So again, we have our diagram. So the muscle is the mailbox. And the acetylcholine receptor is shown the same with the arrow going through it, but there's these other molecules that live near it and they help it. And one of them is called MuSK. And patients with MuSK myasthenia gravis will often look like patients with acetylcholine receptor myasthenia gravis, but the disease in terms of what's happening molecularly here is actually very, very different. And it responds very, very differently to therapeutics, which again, I'll reiterate later, but the point is that if you don't know your antibody subtype, it's important that you try to find that out, and it will help inform you about medications that will work or won't work in particular forms. So the theme again is autoimmunity, and what we see here is this. In MuSK myasthenia, patients are again making autoantibodies that are binding to the muscle, and they're interfering with the way that the muscle receives the signal in a very efficient way. And I'm going to show you that on the next slide here. This is a diagram. It's a complex diagram, but the one on the left side is more simple. In order for the muscle to receive the signal efficiently, you don't need one mailbox, you need a lot of mailboxes, and they all need to be together. And so MuSK tells the muscle, put all your mailboxes together. I want them all in one place. And the antibodies that these patients will make will interfere with that clustering or collection of mailboxes. It keeps them all spread out, and that's an inefficient way to signal. So you see on the left side, I'm showing you that there's a whole bunch of mailboxes, and then you can deliver the mail very efficiently if they're all clustered together. And that's what's happening here. And I wanted to, now we have a clicker going slowly. There we go. I wanted to show you something from the lab that I think you'll find interesting, or hope you'll find interesting, is that we can actually see this happen. And so it's simply looking at the muscle under a microscope and asking if the mailboxes, which is the acetylcholine receptor, are spread out or clustered all together. And if you look at the top right, where I have the little box, the yellow box that says ACHR clustering, that's mailboxes clustering. All those little tiny green dots, not the green background, the little green dots are the acetylcholine receptor or the mailboxes all clustered together. And then on the bottom left now where there's a blue antibody, the blue Y-shaped molecule, you'll see that those clusters are much, much fewer. They're almost gone. That's a real antibody from a real patient that is interfering with the clustering of the mailboxes. And that's how MuSK myasthenia is generated. Very different from what I just showed you with ACHR. In ACHR, you're getting an antibody that can block the message from being delivered, or an antibody that can activate another part of the immune system that damages tissue. Here, it's stopping efficient signaling by this clustering process. Very, very different, and they respond different to therapeutics. And then the last autoimmune part that I wanted to mention was this very rare form called LEMS. It's Lambert-Eaton myasthenic syndrome, and here, it's a situation where now the antibody, again, in the middle, we're back to our home base. In the middle, you'll see a purple antibody, the purple Y-shaped molecule. It is binding to the nerve this time. The three others I just showed you, the two examples for acetylcholine were on the muscle, and then MuSK is on the muscle. So that's the mailbox. We're attacking the receipt of the signal. Here, you're attacking the delivery of the signal. So the immune system now is stopping the post person from actually delivering the letter at all. And that's the same type of interruption. You're not going to get a signal transmitted if there's no one to deliver the mail. And so it's another form of the disease that is an example of how there's so much heterogeneity underlying this disease. There's so many different flavors. Okay, a very important type of myasthenia gravis is called seronegative. Very, very important. It's very important because we don't know a lot about it, and that impacts patients and their caregivers. And so in seronegative, I'm going to use the home base again to show you the nerve, the post person, the envelope, and the mailbox. We know there's a disruption there. It happens here. It happens at the neuromuscular junction, but we don't know why. Is it antibodies? Is it our immune system again? Or is it something that's genetic? It's not really clear. And that's something that we're working on very hard because it's very important to understand this in order to treat it properly. So what are some of the things that we think might be happening from the research side in seronegative myasthenia? One may be that these patients have antibodies that I mentioned before, just like patients with AChR myasthenia or MuSK myasthenia, but we can't detect them. And that's where the name seronegative came from. Folks have myasthenia unquestionably, but when they run these tests, they can't actually see whether or not the antibodies are there. But they may indeed be there, they just may be below the level of detection. So that's one possibility that we're exploring. Another is that I told you there's a genetic form of myasthenia. And it may be possible that some patients, some with seronegative myasthenia, do indeed have a genetic form that has not been identified yet. So that's a possibility that is being explored by research. Another possibility is that there may be antibodies. It's an autoimmune disease, but the particular target has not been identified. So is it attacking something? Is it attacking the post person? Is it attacking the mailbox? We don't know, but that's what we need to figure out. And I'll just highlight, and I put it in blue, I promise you that active research is taking place on this. The field of investigators that work in myasthenia are very interested in understanding seronegative, including my lab. We're working on this now. Okay, so the last part for this is a congenital form. I wanted to tell you a little bit about that. Very different. This is not autoimmune, no immune system involvement here. This is genetic. And so these are called the congenital myasthenic syndromes because it encompasses a large group of patients that have this disruption in the neuromuscular junction. And it's due to a gene change. It's genetic. And a gene change can influence the way the neuromuscular junction works. And so back to our home base, very simple. Well, what can go wrong in CMS? Anything in this system that is either the delivery of the message or the receipt of the message can be disrupted by a genetic mutation that changes it enough so that it's not efficient at signaling. And so I've given some examples here of how perhaps acetylcholine, the envelope that's being delivered, the mail that's being delivered, can't be produced very well. So that could cause a type of CMS. And there's many different flavors, and I'll show you how many there are. This is how many there are. Individuals who usually have just one genetic mutation, and 2 CMS patients may have different genetic mutations between them. It's not always the same. On the right side, I show you that 35 genes have actually been identified to date, all different genes in different individuals that have been identified that are the cause of CMS. So there's quite a bit of heterogeneity there as well. And the types of treatments that are applied to patients that have the autoimmune type would not respond if they have CMS. So it's a very different category of disease that has to be treated much differently. And then in the last part here, I want to talk a little bit about how we can target any of these mechanisms therapeutically. And I think what I hope this highlights for you is that when you understand the fundamental mechanism, when you understand how it works, you have an opportunity to bring a therapeutic to the patients that will help them. And so we're back to our diagram again. And on the right side, we're looking at the neuromuscular junction. And on the left side, what I've showed you is the Y-shaped molecules. Those are antibodies that cause the disease. And there's a special type of cells that produce them. They're just called B cells. B as in boy. So B cells make antibodies, and the antibodies will cause different types of myasthenia gravis. So that's the system that we just went through. This is a simple diagram demonstrating what happens. So how can you target that therapeutically? It's complicated, but it's also straightforward. So I've made it straightforward. How about this? Why don't we remove the antibodies? If the antibodies cause the disease when they bind to the neuromuscular junction, take away the antibodies. You can do that. And so there's therapeutics that are available now that will actually reduce the number of antibodies that an individual has within them so that you don't have as many antibodies attacking the neuromuscular junction. And that results in improvement in the disease. So this therapeutic works. And again, what I'm highlighting as a scientist, not a clinician, is that when you understand this process, it's easy to figure out how to target it. And that's what's happened here. Remove the antibodies, and that can be effective for some patients. Another way is I showed you complement earlier. And complement is this other part of the immune system that's very important for damaging tissue. Well, what if we inhibit complement? What if we slow down complement? There's therapeutics available that actually will decrease the ability of complement to be activated. And those therapeutics actually work quite well in many patients, not all, many patients with myasthenia gravis. So again, understanding the system, understanding that complement's an important part, allows the treatment to be successful to the right and delivered to the right patients. And then finally, you can even go further back in the process and remove the cells that make antibodies. What if we just took away B cells and then you can't make antibodies, and then you're not going to have antibodies that are binding to the neuromuscular junction and causing disease? Well, that works too. And in some patients, if you remove their B cells, they will not make the antibodies that cause the disease and they will get better. And this is the highlight that I wanted to make, is that in patients with MuSK myasthenia, there's particular therapeutics that will remove the B cells, and these patients will go into a very long, stable remission for years. And it's very, very effective. These are MuSK patients. MuSK patients respond to this, a very particular type of removal of B cells very well. The same is not true for patients with AChR myasthenia. Some of them respond, some of them don't. And again, what I was trying to highlight is that knowing your antibody subtype and having an understanding of it and having a basic understanding of these processes is very important for understanding your care. And so for this first part, the other two parts are coming up in their smaller vignettes. The take-home messages are as follows. The neuromuscular junction, the NMJ, is where the disease activity is found in all the different types of myasthenia. There are several subtypes, and there are myasthenic syndromes as well. There's the congenital myasthenic syndrome, which is genetic, and the autoimmune forms of myasthenia are defined by the type of antibody that binds to the particular protein at the neuromuscular junction. So it's defined by the antibody type. Acetylcholine receptor, muscle-specific kinase, that's MuSK. Seronegative, we don't know. LRP4 is very similar to AChR, and then the Lambert-Eaton that I mentioned. And then finally, as I pointed out in the last couple slides, the therapeutics that effectively target this dysfunction are now used and many more in development, and it's because of this understanding. Okay, I'm looking at my timer. I'm good on time. And before we go into the two vignettes, we're going to take a break, an interlude. So the interlude is a little history. If you're interested in history, I encourage you to read about Dr. Mary Walker. So Dr. Walker is shown here on the left side of the slide. She was a physician working in London in 1935. And she had patients with myasthenia gravis that she was unable to treat. And what she did was think that if I could increase the signal that's coming from the nerve to the muscle, these patients might get better. And she actually wrote a letter to a chemical company and asked them to make her a compound, a chemical, that would enhance the amount of message being delivered to the muscle. And it's called pyrostigmine. Some of you might have heard of that. And it works. And she did this in 1935, and we're almost 100 years on from that, and it's still used to treat myasthenia. So a remarkable woman in the history of myasthenia gravis. So that's our interlude. Okay, 2 small vignettes. The next one, number 2. This is the first of the 2 last small vignettes. How academic research works. I think it's very important, particularly in this time, to discuss the landscape of academic research very briefly from the perspective of a scientist that performs academic research to benefit all of the folks in this room. So this is what academic research looks like. I showed you this earlier. This is my team on the bottom left and our laboratory in New Haven, Connecticut, and all of the fine folks that are working in the lab. These are actual real pictures of them doing real Myasthenia gravis research that I took just last week. I want to tell you a little bit about how does it work? How does academic research work? I told you I don't teach. I spend my time running a lab. The lab is run a bit like a business. And so I want to, on the left side, what I've shown you is what are the expenses that are required to run a lab? And one of the most surprising ones is the first one. It's the salary and the fringe benefits for all of the people in the lab. Everyone in the laboratory has to be paid, of course. But the university doesn't pay that. No universities pay the full salaries of all the folks that work in labs or the professors that run labs. They don't. It's not the way it works. And then there's all the laboratory reagents, the consumables, the plastics, the equipment, and then publishing in meetings. So it's an expensive endeavor. Where does the money come from? So at the top, in the middle, I'm showing you where the money comes from. Well, obviously, we're here at the MGFA, so it comes from places like the MGFA. Sam had mentioned that there's been tremendous amount of research support by the MGFA, and that's wonderful and important and essential. It also comes, and the lion's share of it comes from the US government through the National Institutes of Health, which is us, the American taxpayers. So it's our money that's being distributed by the NIH to the universities for this research to take place. And the last point I want to make is that when the government gives research money to universities like my lab to do research in myasthenia, it's given in 2 parts. On the left side, I'm showing you the first part that I just mentioned, the laboratory, the salary, and everything we need to run the lab. On the right side, the second part of the funding, and they come together, it comes in 2 parts, is money that goes for the university to provide infrastructure. There has to be buildings, there has to be administration, legal teams, human resources, huge amount of regulatory and compliance issues that need to be handled, lights, heat, landscaping, all the things that are necessary to run a business. So these are the 2 parts of funding. This is how funding works. Importantly, the funding is never a handout. It is not a handout. It is highly competitive to get grants to run a research laboratory. And this is a graph from the NIH showing you what we all know as faculty members doing research, that 88% to 90% of submitted grants do not get funded. It's that competitive. Only 10 to 12% of grants get funded. You have to keep trying, you have to keep getting better. Is this a bad thing? No, it's a good thing because the best science gets funded. The best science is supposed to help people who are sick. And so this is a good thing. So it's not a handout. And then the last part that I want to point out is what's happening now? Are things changing? And yes, things are changing. What's being proposed is that instead of the part 2 of the funding that goes to the university given at the percent necessary to do the research that we do, there has been a proposal that from now on it's 15%, that's it. But the rates are actually closer to 50 to 70%. That's how much is needed for the infrastructure for part 2. So at 15%, it's not sustainable for any university in this country, regardless of where they are, to continue to do the research that they're doing in the way that they're doing it. So this is impactful to all of us in this room. And then the last part, I want to make a point in this particular disease called neuromyelitis optica. And I've picked this disease because it's a very, very lucid example of why this is important. Neuromyelitis optica is a disease that looks a lot like acetylcholine receptor myasthenia gravis. It's caused by antibodies that I just talked about, and it sets a paradigm for how important research is. So on the left side, I want to show you in 2005, neuromyelitis optica antibodies were discovered by Vonda Lennon at the Mayo Clinic. And that was a major discovery because she defined this new disease, NMO. Several years later, it was discovered that complement, this other part of the immune system I talked about earlier, complement, is key in mediating the disease. And so there's now an understanding of how the disease works. And then in 2014, a diagnostic blood test was developed so that these patients could be identified. These patients often look like patients with multiple sclerosis, and so differentiating them is very important for treatment. And then finally, in 2019, as I mentioned before for AChR, a therapeutic complement inhibitor, that was a medicine, was provided to these patients, and their very, very difficult relapses that can put them in a wheelchair if left unattended stopped happening. And so the therapeutic complement inhibitors worked. The important takeaway from this is that the left side of this figure, panel 1, panel 2, and panel 3, are all academic research funded by the NIH. The fourth panel is the pharmaceutical companies, and they developed the drug and took it to market and tested it to make sure that it worked. And so there's a partnership between the universities and the drug companies to bring this to bear. And it's an essential partnership, and the concern is that the left side may be diminished, and I think we all need to be concerned about that. So that's that vignette. And now I'll go on to the very last part where I want to share with you some examples and a little bit of detail of some of the very exciting work that's going on that the MGFA actually funds very directly, that is related to what I just explained to you about the way myasthenia works. So as Sam mentioned, there's been a great deal of activity in terms of funding from the MGFA. We have pilot awards available to investigators that provide seed money for projects to start. We have larger awards named after Nancy Law, the former CEO of the MGFA, that are for more senior investigators to continue big projects in myasthenia. And we have this wonderful postdoctoral fellowship called the McSpadden Award. And this is a very important award that captures young people who are starting their careers in research, and it gives them funding for 3 years to work specifically on myasthenia gravis. And that usually captures them to stay in the field, and they stay in myasthenia and keep working on that. It's very important to do. And we've expanded this to beyond the US borders to include international investigators. And I want to point out the reason that we did that is because we want the best science. The best science can happen anywhere in the world. It can happen here, and it can happen in Europe, Japan, Asia as well. So we're open to investigators from all over the world to provide the best science. Here's a couple quick examples of them. One of the most important things that the Myasthenia Gravis Foundation did in the past was develop what's called the MG composite score. And if you look at the little diagram I made, you can see there's different classes in the way that patients can be rated in terms of their disability. So class 1 is doing quite well. We're all the way over to the right. You can see that the classes go down with increasing debility and fatigability. This is really important because it's a way to measure whether patients are responding to a therapeutic. If a patient comes into a clinician's office, and my colleagues will speak about this more today, I'm sure, they may be class 3. And then you give them a therapeutic, you have to know if they're getting better. Well, you have to have some sort of measurement tool. This is the measurement tool that allows you to determine whether somebody's doing better or getting worse. It's obvious, but it didn't exist a number of years ago, and the MGFA was behind it. I told you earlier that antibodies can fit into the lock and key at the neuromuscular junction and block the mail from being delivered because it fits perfectly. My good friends and colleagues at the University of University of California in San Diego have used this very powerful molecular microscope to actually show what I just explained to you, that antibodies can actually stick inside of the spot where acetylcholine, the message, is supposed to be delivered, and block it. It's never been demonstrated before, and with this very powerful microscope they demonstrated it, and the MGFA supported their work. Ricardo Maselli is at UC Davis, and he works on congenital myasthenic syndrome, and his work is very interesting and important for two reasons. One, he's trying to define new types of congenital myasthenic syndrome, and so one of the ones he's working on, he's trying to understand why the nerve can't deliver the message. And he's finding out that it can't put the mail into an envelope, if you will, and then package it to go across the space for the muscle to receive. So in addition to defining how the disease works, he's also developing therapeutics that can repair the gene that's damaged in patients with this type of CMS. And then finally, my good friend Amy Payne, who was formerly at Penn and now at Columbia in New York, is also working on a therapeutic that I hope has some understanding here after I had showed you the way that the mechanisms work. Again, on the left side, I'm showing you that B cells are making the Y-shaped antibodies, and the antibodies cause the disease. So Amy's group has developed cells, very specific engineered cells, that will kill the B cells that make the antibodies that cause the disease. So I had mentioned earlier that one of the ways to remove antibodies is to kill the B cells. How do you kill the B cells? Amy's developed a way to do it with very precise targeting to remove only those B cells that produce antibodies in patients with myasthenia. Again, MGFA supported. She's a Nancy Law Award recipient. So my time is up. I'm right on time, Sam. Very good. So with some time for questions. And so I'll acknowledge, you guys have to let me put the acknowledgement slide up. It's very important. Can you go back? You can't skip all the folks that did the work. So I'll just say with the slides that are coming up, that I thank you for that. This is my team at Yale who spend very long days working very hard in the lab, trying to understand this disease. They're very dedicated young people, and I owe a great deal of gratitude to them. And thank you. That's for them. Thank you. They deserve it. And then one more slide. If you, maybe you guys are controlling it. Forward me one, please. It's the funding. And so, oh, where are we going? I'm not touching it. The very last slide, please. It's the funding. I'm just going to say it. The NIH supports me, so that's very important. The MGFA supports me, that's very important. Here they are. I've been very fortunate to receive awards in terms of grants from Conquer MG, the Muscular Dystrophy Association, MGNet, my friend Esther Land at the Myasthenia Gravis Foundation of Michigan, the Neuromuscular Study Group, Friends at InfuCare. I told you that there was a partnership between academic labs and industry, and you can see that I've worked with Argenx and Seismic and Amgen to help develop better therapeutics to target the right patients the right way. And of course, private generosity from very generous donors, the McSpaddens, Dr. Martin Schuberg and Ann Sweeney. I thank you all very, very much for your kind attention. I hope this was valuable for all of you, and I look forward to questions and meeting you all as our meeting continues over the next couple of days. Thank you. There are microphones, I understand, and maybe a few questions from Slido, so I'm waiting to hear. Okay. Okay. This is what Kelly, no, I'm going to make you do it. Kelly, please, come on up. We're a community here. So I'm a scientist. I'm not a clinician. I don't treat patients. I see a lot of the questions are about therapeutics. And Kelly is a dear friend of mine and can answer some of these questions better than I can. We'll do it together, please.
Thank you. I'm the token clinician here. So, so first of all, okay. Well, yeah, first one I'll take from the top. Can someone have more than one type of MG? And I think Kevin could answer this as well. But yes, there are such patients that are double seropositive and might have MuSK and acetylcholine receptor antibody mediated myasthenia. Not a ton, but they are reported in the literature. In terms of holistic therapies, I'm probably not going to touch that up here in front of everybody, just in terms of talking about off-label treatments and anecdotal evidence. In terms of treatment options available to the seronegative community in the future, I can speak to the fact that there have been some recently conducted clinical trials and starting ongoing clinical trials that really do include and target the seronegative patient population. And so I'm extremely optimistic by the progress there. Okay, number 4 is you. Yes.
So number 4, I'll read it out loud. Has there been any significant research into causes of MG? What are the reasons for autoimmune disease? That's a great question. And in all of the autoimmune diseases that I mentioned today, you know, MS, type 1 diabetes, the cause is at the root of the problem, exactly right. And so researchers and investigators worldwide are looking to find what is the root. I think the simplest answer, and there isn't one simple answer, but it's typically a constellation of a number of things. There has to be some genetic predisposition. There has to be perhaps some exposure to environmental antigens like particular infections. So I know this is unsatisfying. It is complicated and we don't know for sure. But the last thing I'll say is that when I told you that the immune system was not functioning the way that it should by attacking self, your immune system is supposed to be taught what's called tolerance. It's when your immune system develops, it knows what's you and what's not you. And in autoimmune disease, that goes off the rails. And that's the basic cause of autoimmunity, that there's a failure of what we call self-tolerance. There's other factors that contribute, but that's what it is.
There's a good question. I mean, maybe I just think that question about the ocular MG therapeutic, I mean, I know we have a talk on therapeutics in a moment, and so I certainly don't want to steal anyone's thunder. But I think there was a question about why the treatment recommendations are different in ocular MG versus generalized. And I'll be honest, my impression is that a lot of these newer therapeutics have simply not been trialed in ocular MG. And so these newer therapeutics at this time are not labeled to treat ocular MG. Similar to the question about the thymectomy, it hasn't, you know, that study was done in generalized myasthenia. Do you want?
How about the Musk presentation, the worst presentation of Musk, Deirdre at the bottom?
Quantified. Okay. So I've seen references to Musk MG having a worse presentation than AChR positive MG. Never seen this quantified. Can you please speak to this? I mean, again, I can just speak to sort of the general body of literature, but I'm not going to reference a specific study that compares the two. But you're right, Musk MG patients tend to have a different course. They often will have a higher rate of going into crisis, right? I think it's about 30+% go into crisis compared to, and meaning you get intubated, ventilated in the hospital, not just an exacerbation, in contrast to AChR positive, which is closer to 20% or so. And they can be more treatment refractory. And to the point that Dr. O'Connor made, the therapeutics are different in Musk MG. And so I think for many decades, we were using some of our AChR, you know, more appropriate therapeutics in our Musk population, and they weren't responding. And I think we've gotten a lot smarter recently. And so I'm very optimistic about the treatment outcomes in Musk right now. Sure.
Yeah, and there's another one from Ben Crowther. Thanks, Ben. What is the relationship between the thymus and myasthenia gravis closer than that with other autoimmune diseases? Indeed, the thymus is very important in particular subtypes of myasthenia gravis, and our clinical colleagues will be speaking about that today in that particular subtype. But from the immunological perspective, it seems that in some patients with early onset myasthenia gravis, another subtype you'll hear about today, the thymus is indeed involved, and a lot of the immune activity can be observed in the thymus, and which is why thymectomies are performed to remove that activated immunology that's contributing to the autoimmunity. It's not a perfect treatment, but it's very helpful. And it is true that in myasthenia, the thymus is directly involved in a subset of patients where it's not observed in other autoimmune diseases. So there's a clear connection there.
I think that's an intriguing question. I don't know, the one in green.
Everyone, one more question, only one more.
Oh, you can take that, all
I
right?
mean, I
Yeah,
can
we can
speak
both
to it,
take it.
yeah.
Go ahead, take it. That's a good one.
So it's a good question. So if you were AChR positive in the past, become negative when you're in remission, or will it always show up in your blood test? And I've got to be honest, for the few patients I have that went into remission off of drug, I don't routinely check it longitudinally, clinically, because that really isn't standard of care. But of course, we do see the antibody level go down with these therapeutics, right? Some of the ones that you mentioned, the FcRn inhibitors, as well as plasma exchange, will certainly drop those levels in keeping with some disease improvement. But in terms of that small population that completely goes into remission off of drug, do you know if their antibodies completely disappear?
So it's a good question, and I think that they can disappear for a sustained period of time, but I don't think they'll go away permanently. There is not a cure for autoimmunity, and so eventually they will come back, but that involves management. You have to make sure to check to see if they're coming back, and if they do, there needs to be treatment again. But I don't think that our current therapeutic landscape offers a reset for our immune system.
All right.
We have time for one or two more questions, and that's it.
So Mike, good that there's a gentleman there. Yes, but you should take that.
Sure. Yes, they're available panels.
Can you repeat the question?
Yeah, oh, I'm sorry. I'll repeat it. Is there any commercially available genetic testing for congenital myasthenic syndromes? And the answer is absolutely there is. Yes. So we send that commonly in patients who are not, I mean, to your point, responding to typical treatments, and our suspicion is high. Sorry. Yes. Oh, hi.
Hey, how you doing? I was just wondering, with the new research and developments that have been done, like someone who was diagnosed 10 years ago, 20 years ago with one type of myasthenia, does it make sense to get retested because development and new research has been done over that time, and maybe they haven't had that test wasn't available at the time? Does it make sense to be retested and see if treatment and therapy needs to be changed for that person?
Well, I think, please correct me if I'm wrong in understanding the question. So you're asking whether or not someone should be retested for maybe their specific antibody type over time. Is that correct?
Right.
Yes.
Specifically? So there are a percentage of patients who are initially seronegative. This is an excellent question, that then flip positive. And so most of us who are very active in this space will repeat AChR antibodies every 6 to 12 months after someone is initially seronegative. And so yes, that does have clinical implications because it opens up the door to additional therapeutics. It's a great question.
We need to get a mic. I'm sorry, Mike.
Or I can repeat it.
We do have a minute left, just 1 minute.
Okay. Oh, okay. In the patient community, the patients that often struggle the most with getting a diagnosis or being diagnosed with MG are those that you spoke so confidently about, the antibodies and the ones that are diagnostic for MG, and you included LRP4. And what I see often in the patient community is right now, at least, with regard to trying to get diagnosed in clinic by providers, that seems to be the most challenging. And I just wanted to know if there's anything moving forward, if among providers, that's going to be something that there's going to be a change moving forward.
I'm sorry, for ocular MG specifically? Oh, LRP4? So I'm sorry if I misunderstood it. Yes, there's a commercially available test for LRP4, but for those patients who clearly have myasthenia gravis based off of electrodiagnostic testing. I mean, often I don't go and check LRP4 because it doesn't change the treatment paradigm at this current moment, because we would use the same kind of armamentarium of therapeutics for seronegative as we do for LRP4. Okay, I think we're out of time, so I'm happy to address that afterwards. Yeah, thank you.
Okay. Thank you, Kelly. Thanks, Kelly, for helping.
Good morning, everyone. My name is Daryl Lee, and I serve as the National Director of Finance at MGFA. I want to make a little bit of a housekeeping point. After our next speaker, we will have a 5-minute coffee break within this building. Okay, we're doing our best to try to get back on time since we're running a little bit late. Okay, just wanted to kind of point that out. It is my honor to introduce our next speaker, Dr. Suraj Muley. Dr. Muley is the Medical Director of Neurology at the Bove Neuroscience Institute in Scottsdale, Arizona. Dr. Muley completed his residency at Neuromuscular and Clinical Neurophysiology Fellowship at the University of Minnesota. He specializes in immune-mediated neuromuscular disorders. He holds a faculty position as Professor of Neurology at the University of Arizona Medical School. He is a fellow of the American Academy of Neurology, American College of Physicians, and Royal College of Physicians in the UK, and a Fulbright Senior Scholar. Please welcome Dr. Muley.
Right. Thanks for that introduction. So I'm going to switch gears a little bit. I'm a clinician, and I'll talk a little bit about myasthenia gravis, the disease, and then go into the treatment of the disease, which is, to me, the most exciting kind of part, because, you know, I trained in the 90s, and we were taught to treat myasthenia a certain way, and we did that for a long time. And a lot of how physicians kind of treat any disease is dependent on how they're trained and what they've done. And, you know, I started the myasthenia clinic in Minnesota in 1999. So for 20 years, I treated myasthenia a certain way. And in 2019, we had the first FDA-approved treatment with eculizumab. And the question was, how does that fit into the treatment algorithm? And then we have FcRn inhibitors, which again change the landscape. And we will have B-cell depleting therapies soon and so on. So the question to me is, what's the best way we should treat myasthenia for the patient, both in the short term and the long term? I think a lot of the clinical trials that are done are looking at short-term improvement in symptoms, which is over, say, 6 months, and maybe sustained improvement over time. But that's really the question to me. What is the best way to treat myasthenia? And I'm still not completely clear in my mind. I have my ideas, and we'll talk a little bit about that. So we'll start with talking about the disease first. I hope I can get this to work. All right. I'm advancing them, right? Okay. All right, so this is, Dr. O'Connor talked about this, but this is really the neuromuscular junction here. This is the nerve, and then this is the muscle. When the nerve is activated, it releases acetylcholine that then binds to these receptors on the muscle membrane, which leads to muscle contraction. And the number of these receptors on the muscle membrane really dictates how well the muscle contracts, essentially. And in myasthenia, the number of receptors on the muscle membrane is reduced, so the force of the contraction is reduced, and there is fatigability as well. Now, the antibodies that are responsible for myasthenia do three different things to these receptors. One is they bind to the receptors and don't allow the acetylcholine to bind to the receptor. The second is they cross-link the receptors, which leads to internalization and degradation of the receptors. And third is they activate complement, which is a protein that fights encapsulated bacterial infections, but it's also activated in this disease, and that also damages the receptor. And then over time, there is also simplification of the postsynaptic muscle membrane. So the membrane becomes flatter over time. So the surface area of the muscle membrane becomes less. And remember, these receptors are not a fixed structure on the muscle membrane. They are recycled with a half-life of 20 days. So we are reforming these receptors, all of us, with a half-life of 20 days. And in myasthenics, the half-life is reduced to around 10 days or so because of mRNA upregulation. So if you prevent damage of the receptor, new receptors can form quickly, relatively quickly, and you can improve relatively quickly. But when there is flattening of the muscle membrane, there is not enough surface area for the receptors to form. So in some patients, you can get fixed weakness over time. So some patients, despite all possible treatments, can have mild ptosis, mild diplopia, and so on, which is irreversible. And that's partially related to end-plate myopathy. And the bigger point then is to make sure that we treat myasthenia appropriately, optimally early on, so that you don't get damage of the postsynaptic muscle membrane. So, the first step in diagnosis is, is the clinician seeing this patient. Maybe the PCP first sees a patient with fatigue, maybe ptosis. A lot of patients land up getting an MRI to make sure it's not a stroke, for example. But these are the common clinical features which all of us need to recognize so that the diagnosis is not delayed. Because you can do a blood test only once you first suspect the diagnosis. You can't do the blood test if you're not thinking about this diagnosis. So fatigable weakness is a characteristic first symptom in myasthenia. And when we say fatigable, what it means is that the weakness gets better with rest. So most patients wake up in the morning, they're fine. As the day progresses, they have weakness, symptoms of ptosis or droopiness of eyelids, double vision, and so on. And the symptoms can get better with rest. And that's what we call fatigable muscle weakness. Over time, the weakness can become fixed if the disease is not treated. And ocular symptoms tend to be the common initial symptoms. So, again, ptosis and diplopia are common. Difficulty with speech, chewing, swallowing is common. Difficulty with breathing, difficulty with arm and leg function, and so on. And again, these symptoms overlap with other conditions like multiple sclerosis, strokes, cranial neuropathies, and so on. So the diagnosis can be delayed if the physician is not thinking about this disease. And then sometimes the antibodies can help with diagnosis. At the bedside, you can do a tensilon test where you're basically blocking the degradation of acetylcholine, which can improve symptoms transiently. You can put an ice pack on an eyelid that is drooping and look for improvement, which can also help you at the bedside. But the antibody test is really definitive in the clinical context to make a diagnosis of myasthenia. And in patients with generalized myasthenia, 85% of patients will have antibodies. If patients have ocular myasthenia, 65% of patients will have antibodies. So sometimes in ocular myasthenia, you may not see an antibody positivity, and the diagnosis is based on other kind of tests. So like I said, in generalized myasthenia, 85% of patients will have antibodies to acetylcholine receptor. In ocular myasthenia, 65% or so patients will have antibodies. MuSK antibody is positive in 40% of the seronegative patients. And then 1 to 2% of patients can have LRP4 antibodies. I've seen that a lot of patients with myasthenia also have thyroid disease. So hypothyroidism is common. Some patients can have thyroglobulin and thyroid peroxidase antibodies, which can cause ocular dysmotility. And you can sometimes see a combination of ocular myasthenia and ocular thyroid eye disease, which can complicate management and response to treatment. We always do a CT scan of the chest because a thymoma is the only real known cause of myasthenia. In most patients, even the ones that don't have a thymoma, I think thymic dysregulation of autoimmunity is probably the cause of myasthenia, although in MuSK patients, the thymus tends to look normal. And then we talked about the IcePack test and the Tensilon test, which can support the diagnosis as well. And then electrodiagnosis can be useful. In electrodiagnosis, we do repetitive nerve stimulation to see whether the response of the muscle fatigues with that. And we can do a single fiber EMG, which should be abnormal in 99% of patients with a neuromuscular junction disorder. But those tests still don't prove that it's an autoimmune disease, right? All they tell you is that there is a neuromuscular junction defect, but they don't tell you that it's related to, say, an autoimmune problem, because congenital myasthenic syndromes will also have abnormal single fiber EMG, for example. Lambert-Eaton myasthenic syndrome will also have abnormal EMG, single fiber EMG. So that it still supports the diagnosis, but response to immunotherapy is what then proves that it's an autoimmune kind of process. So we classify myasthenia as early onset, which is before the age of 50. Late onset is after the age of 50. In early onset myasthenia, thymic hyperplasia is common. And removing the thymus gland can help patients with early onset myasthenia. Here, the cutoff for late onset is 50, but the thymectomy study that was done was between the ages of 18 and 65. So, and they showed that thymectomy can be useful in even patients without a thymoma because it can, improve your immune system in the sense that it'll fix the dysregulation in the thymus. Now, thymoma is common in myasthenics. MuSK patients tend to have a normal thymus, so removing the thymus gland is not that beneficial in patients with MuSK myasthenia. LRP4 is a rare disease. Again, I don't think we have enough information about whether thymic changes are seen in LRP4 disease. And in seronegative disease, it's very difficult because we don't, the first step is we don't even sometimes feel confident that it's an autoimmune disease. And if it is an autoimmune disease, we don't know what the precise immune mechanism is. So we don't know whether the thymus plays a role in seronegative myasthenia. And then ocular myasthenia to me is just a mild form of generalized myasthenia. It's where the symptoms start in the eyes. In majority of patients over the first 2 years, symptoms generalize and patients develop generalized myasthenia. So there are some coexisting conditions that are common with myasthenia. We talked about thymoma. Other autoimmune diseases can coexist in patients with myasthenia. Patients can develop complications from treatments like diabetes, hypertension, if they're on steroids, osteoporosis, and so on. So we have to think about coexisting diseases as well in myasthenics. So coming to treatment options, I think that this is a list of all the treatments available in myasthenia. So it's, I think the more difficult question is how do you treat an individual myasthenics? We talked about the clinical features of myasthenia, and even though there is a limited number of muscles that become symptomatic in myasthenia, each patient is a little bit different in terms of the tempo of disease, how severe the disease is, which specific muscle is more severely affected in a particular patient. And that really makes the treatment very individualized. So I think that there is no standard formula for treating a myasthenic patient, I think. But there are certain rules and algorithms that we are trying to develop that would have general applications for most patients. So these are the treatment options. First is thymectomy, which is removing the thymus gland, which all patients with a thymoma really need. If patients don't have a thymoma, it's still a valid treatment for some patients, especially younger patients. Glucocorticoids or steroids were the first line treatment for a long time and are effective in myasthenia. The problem is adverse effects, and we'll talk about pros and cons of each of these treatments in a little bit. Intravenous immunoglobulin is an off-label treatment for myasthenia, but kind of makes sense because it does impact the immune system in multiple ways. It decreases T and B lymphocyte activation. It has an impact on complement. It has an impact on FcRn receptors. And so, intellectually, it makes sense that it should work, even though the trials done with IVIG were not positive for myasthenia. So it's not an FDA-approved treatment, but it can be an option for patients, especially patients who are immunosuppressed, because unlike other treatments, IVIG does not suppress the immune system. And in that sense, can be a valid option for patients who cannot tolerate immunosuppression, for example. Plasma exchange is generally used in a crisis situation where patients are in the hospital with severe dysphagia or difficulty swallowing or difficulty breathing, and can be very effective. But it's generally not useful in the outpatient setting because it's tough to have a central line going to the plasma exchange center frequently and so on. So makes it challenging to do plasma exchange in an outpatient setting. 10 years ago, I had patients who would get outpatient plasma exchange chronically, but now with newer therapies, the need for outpatient chronic plasma exchange has reduced. And again, comes with complications, line infections, blood clots, fluid imbalances, and so on. And long-term immunosuppressants have been used for a long time in myasthenia. The only trial that was done with mycophenolate was negative for myasthenia. And despite that negative trial, people have used long-term immunosuppressants in the hope that they can allow you to decrease the dose of prednisone over time. But with newer treatments, we are still trying to decide what the role of these long-term immunosuppressants should be. Should we use these treatments early on, or should we move on quickly to the newer treatments, which are very effective, but they work downstream in the immune pathway. So the newer treatments don't decrease the production of antibodies. They block the antibodies from damaging the receptor, but they're not decreasing the production of these antibodies. And so the long-term immunosuppressants have an advantage in the sense that they can decrease the production of antibodies and perhaps induce a remission in the long term. And then B-cell depleting therapies like rituximab are very effective in MuSK patients. I've had patients who are treated with rituximab for MuSK, and they don't see me for a year because there are no symptoms for a year. And then after a few years, they develop symptoms and they come back and see me. So it can be very effective in MuSK myasthenia patients, not universally as effective in acetylcholine receptor antibody patients, although we'll show you some data showing that early treatment with B-cell depleting therapies could influence the course of the disease in some patients. And then complement inhibitors like eculizumab, which was the first FDA-approved treatment in 2019, can be very effective in myasthenia as well. And then we have ravulizumab and zilucoplan. So we have 3 different complement inhibitors in MG, which can be very effective because they work very downstream in the immune pathway and block the formation of the membrane attack complex, which is really the final pathway in terms of receptor damage. And as I said previously, new receptors can form quickly, and myasthenia can improve relatively quickly with complement inhibitors. But complement inhibitors work only if the IgG subtype is 1 and 3. So these antibodies are all IgG or immunoglobulin G, and there's 4 different subtypes of IgG: IgG 1, 2, 3, and 4. And we know that IgG1 and 3 subtype activates complement, and the other subtypes don't activate complement. So MuSK antibodies IgG4 subtype, which don't activate complement. So complement inhibitors are effective in acetylcholine receptor myasthenia and perhaps in LRP4, which is also IgG1 and IgG3, even though we don't have direct studies showing efficacy. The FcRn inhibitors can be attractive because they can work for all IgG subtypes. And because they decrease IgG1, 2, 3, and 4, so they can be useful for both MuSK myasthenia patients or acetylcholine receptor positive patients. And some of the studies have included seronegative patients as well with FcRn and have shown efficacy in seronegative patients, although the studies weren't powered to kind of get approval for seronegative patients, but there was benefit in those studies in seronegative patients with FcRn inhibitors. So the question is, what are our goals in treating MG, right? I mean, what we're trying to do, really. Obviously, we're trying to make patients feel better. You know, their symptoms should go away completely if possible. We know that in patients where symptoms are ongoing, the risk of a crisis, risk of hospitalization is very high. So, and we know that if we control patients' symptoms well, the risk of hospitalization or a myasthenic crisis are reduced significantly. And the treatment should be relatively cost-effective, safe, and tolerable. So treatment burden is important. You know, if they're disruptive with your daily schedules, if they cause side effects, that's not ideal. So we try to find a treatment that works for patients' schedules and otherwise. And then hopefully induce a remission. What we mean by remission is that patients have no symptoms of disease. In an ideal world, we would like to induce an off-treatment remission where the disease is truly gone rather than controlled with medication. So you can use the term on-treatment remission, which is the disease is completely controlled on medication, and an off-treatment remission where you can withdraw the treatment gradually and the patients are still in remission. And one of the questions is, which maybe Dr. O'Connor has some insight on this, is whether if we use treatments that are upstream in the immune pathway, whether our chances of inducing an off-treatment remission over time are higher than if we use treatments that are downstream in the immune pathway, like complement inhibitors, FcRns, and so on, because we're not really decreasing the production of these antibodies. And so that's the question that is not completely clear, whether using treatments that are upstream in the immune pathway give you a higher chance of inducing an off-treatment remission over time. And then lastly, there's a concept of epitope spreading where your acetylcholine receptor becomes more and more damaged over time, and more epitopes within the receptor are exposed to the immune system, making the immune response more and more robust over time and more difficult to control over time. And so that's another goal of treatment, to make sure that we treat the disease early and effectively so that there is not a lot of epitope spreading and the immune response doesn't become more and more robust over time. So those are really the goals of treatment, I think. So this was the treatment paradigm in 2015, which was before we had any complement inhibitors or FcRn inhibitors. And once the diagnosis is confirmed, the first step was to do acetylcholine esterase inhibitors and consider a thymectomy, especially for patients with early onset disease or patients with a thymoma. And if that was sufficient, if patients did better on that, because you're taking the thymus out, which is improving your immune system in a way that can improve myasthenia. So if that is sufficient, or that was sufficient, you don't really need any other treatment. But it's rare in my experience for that to work as the only treatment in generalized myasthenia. So if patients don't get better with that treatment, then the next step was to do prednisone and azathioprine or mycophenolate, for example. And like I said, prednisone is very effective in myasthenia, but the problem is side effects. If you're lucky, some patients with prednisone for, say, a month or two, get to near normal, and you can slowly taper the dose of prednisone. And if you can get down to a dose of, say, 5 milligrams a day, I think that is the easiest way to treat myasthenia, and it works for patients. They are on 1 5 milligram pill every day without a lot of side effects. But that's not universal. A lot of patients don't tolerate the high doses of prednisone, which sometimes you need to use for months, and you get side effects. There is a need to taper prednisone, which then leads to worsening of disease. In some patients, using high doses is not enough, and they still have a lot of symptoms despite prednisone. So even though that is an ideal scenario, it's not a common scenario, and a lot of patients need more than that. The azathioprine, mycophenolate are treatments that take 6, 12 months, 18 months to really work. So they're long-term immunosuppressants. So they're not going to help you right away. And the hope is that they allow you to come down on the dose of prednisone over time. Now, if those treatments didn't work, then you would try a different long-term immunosuppressant like mycophenolate or cyclosporin. And if that didn't work, we would try rituximab, which is a B-cell depleting therapy, which can work in some patients with acetylcholine receptor antibody disease. But the effect is not robust and can be delayed, and it's not predictable. And so those are the only treatments we really had before 2019. And some patients did well. I still have a lot of patients who, with that treatment paradigm, have done well. They're on a very low dose of prednisone. Some of them are here in the audience on very low dose of prednisone, 5 milligrams a day, and CellCept, and then we are slowly tapering the CellCept. So this treatment paradigm can work in some patients, but not all. And many patients, despite these treatments, continue to have a lot of myasthenic symptoms, and there is a need to treat them more effectively. Also, there is a need to decrease the dose of prednisone relatively early on, which we didn't have a choice previously because we had no other treatments. But now we have options where if patients are developing side effects from prednisone, we can use something else so that we can taper down the prednisone faster. So this was the thymectomy study. I'll go through these studies quickly so you understand how each of these treatments were shown to be effective in myasthenia. So this was a thymectomy study which I was part of, which clearly showed that in patients between the ages of 18 and 65, thymectomy decreased the QMG score and also decreased the amount of prednisone that is necessary compared to placebo. So clearly has an impact on the amount of prednisone you need to control disease and improvement of symptoms in myasthenia. But the initial cutoff date actually was 60, even though, because they couldn't recruit enough patients, the age limit increased to 65. But the point is that the younger the patients, the more effective it tends to be. So I generally don't do it in patients over the age of 50 as easily. If patients are closer to 20 to 30, 35, I think I recommend thymectomy can be very effective in some patients. Complement inhibitors, Dr. O'Connor kind of talked about the mechanism of how complement inhibitors work. But when the antibody binds to the receptor, acetylcholine receptor, there is activation of the complement pathway, which eventually forms the membrane attack complex, which is a protein that really damages the acetylcholine receptor. So complement inhibitors block formation of the membrane attack complex and let the receptors reform, and that's really the mechanism of action of complement inhibitors. It's a little bit delayed. So this is the REGAIN study, which led to the approval for eculizumab, which was the first complement inhibitor in myasthenia. and was administered every 2 weeks. You see a significant impact on MG-ADL score, QMG score, the MG composite, and the quality of life score. So significant improvement in all measures of disease. And I still remember in 2019, when this was approved, I had a lot of patients I had been treating, and they were on high doses of prednisone. They were having side effects. We didn't have options. I had a patient actually who could not work effectively and had to use a notepad to communicate. And she would get severe, she might be in this audience, she would get severe dysarthria by 10 a.m. every day. And when I first started this medication in that patient, I was myself wanting to know how that patient would do. And within a month, she was normal. Her speech was normal. She could start yelling at her grandkids the way she wanted. And she could go to Vegas on the Strip and so on. So it was dramatic, I think. And I think that really changed the landscape of myasthenia treatment. And then the ravulizumab is essentially the same mechanism as eculizumab. There's 4 amino acid changes on the molecule, which increases the half-life of ravulizumab or Ultomiris. And it allows for administration every 8 weeks instead of every 2 weeks, which is quite convenient for patients. So 1 hour every 8 weeks is not as disruptive in terms of treatment burden. And again, the Champion MG study showed significant improvement, both in MG-ADL and QMG scores. And so that can also be an effective treatment. And then zilucoplan, which is another complement inhibitor. Again, you see significant improvement in MG-ADL score and MG composite score with zilucoplan as well. Later on, I'll talk a little bit about pros and cons of each treatment. And then the FcRn inhibitors work in a way that decreases the IgG levels, essentially. And in humans, the half-life of IgG is 21 days. And the reason our IgG half-life is 21 days is because we need IgG to fight infections. And when IgG from the blood goes into the endothelial cells surrounding the blood vessels, they are taken up by the endosome, and they bind to a receptor called the FcRn receptor, which prevents their degradation, essentially. But the problem is the myasthenia antibodies are also IgG. So the myasthenia antibodies are also binding to this receptor, and the half-life of myasthenia antibodies is also prolonged because of this receptor. So with FcRn receptors, you're blocking this receptor, and this salvage mechanism for the IgG is blocked. So that leads to degradation of IgG in the endothelial cells. So good IgG also goes down in levels, and myasthenic antibodies also go down with FcRn inhibitors. So that's really how FcRn inhibitors work. It's kind of like plasma exchange, although plasma exchange will wash out other cytokines, not just IgG, but it's similar to what plasma exchange does. So this was our ADAPT+ study, again, looking at effectiveness of FcRn inhibitors, efgartigimod IV in myasthenia, and you see significant improvement in MG-ADL score and MG composites and sustained efficacy over cycles. And these are seronegative patients. I was mentioning that even seronegative patients may improve with FcRn inhibitors, and this is the data in seronegative patients where FcRn inhibitors can work. And I have patients with seronegative myasthenia where FcRn inhibitors like efgartigimod or Rystiggo can be effective. So this is rozanolixizumab or Rystiggo. This is another FcRn inhibitor, and you can see significant improvement in MG-ADL, MG composite, and QMG. Rozanolixizumab leads to approximately 80% reduction in IgG levels. It's a 6-week cycle. Efgartigimod is a 4-week cycle and leads to a 60% or so decrease in in antibody levels. So depending on the patient's disease profile, antibody levels, how severe the disease is, one treatment could be better than the other for that individual patient. And then this is a Swedish study, which is of interest. And this was a Rhinomax study. So what they did is they used rituximab very early in myasthenia. So the inclusion criteria was age over 18, but their disease duration was less than 12 months. So they had very early myasthenia. The QMG score had to be 6. They couldn't have purely ocular MG. They couldn't have thymoma. And they had to be on a dose of prednisone less than 40 milligrams, essentially. And they couldn't have used prior long-term immunosuppressants. So you see here, at week 14, patients treated with rituximab did much better than patients on placebo. And you see at week 48, these 80% of patients were in minimal manifestation state, as against placebo, much lower. So this raises a question of whether early treatment with B-cell depleting therapies could change the course of the disease and could induce a remission potentially in some of these patients. And remember, the B-cell depleting therapies work upstream in the immune pathway, so they're really blocking production of antibodies. The question that is not answered by this is whether after a certain time on rituximab, whether you can stop it or taper down, and whether the disease remains in remission or not. That would be a good question to answer. So again, going through individual treatments and their pros and cons. Steroids are immunosuppressive, so they suppress the immune system. They decrease T cell or T lymphocyte transcription, so they decrease the ability of T cells to fight infections, but they help because they're decreasing the immune attack against the receptor. They're inexpensive. If they work within weeks, so not very quick, but not as delayed as long-term immunosuppressants. They're oral, so it's easy to take. But the problem is adverse effects. Once you're on steroids for more than 1 to 2 months, you will get some osteoporosis, diabetes, hypertension, weight gain, gastritis, mood changes, glaucoma. It goes on and on. So there is a need to decrease the dose over, after a few months. Long-term immunosuppressants take long to work. There's a risk of lymphoma with long-term immunosuppressants. It's not very large, the risk, but there is some risk. I've had one patient in the last 25 years with lymphoma from mycophenolate. Hepatic dysfunction, bone marrow suppression, we monitor labs and we make sure that's not an issue. IVIG, even though it's off-label, it doesn't suppress the immune system. It modulates the immune system. So in patients who are fragile and have a high risk of infections, it could be an attractive option. Again, the studies are negative. I think we've seen some improvement in some patients. The problem can be side effects with IVIG. You get it every 3 weeks or so. The infusion lasts for 4 to 5 hours. You can get flu-like symptoms, blood clots. So there's some side effects with IVIG as well. Plasma exchange we talked about can be very effective, but doing it outpatient is a problem. But it's very effective in the inpatient setting, which can work. And then thymectomy we talked about in younger patients. So in terms of the newer treatments, so we have 2 different kind of treatment mechanisms. One is complement inhibitors. We talked about 3 of them: eculizumab, ravulizumab, and zilucoplan, and they all block complement. The advantage is that they're rapid in their onset of action. So within a week or 2, patients get better. Not a lot of adverse effects as long as you have meningococcal vaccine. I've not seen significant adverse effects, although you can get abdominal pain, nausea, and so on. In the trials, I have not myself seen a lot of adverse effects. Ravulizumab is every 8 weeks, so it's fairly convenient. But in some patients, there's wearing off. Not commonly, but can happen, where the effect at 6 weeks is kind of wearing off. And in those patients using something every day like zilucoplan, which is an everyday self-administered injection, can be beneficial, and because you don't see the wearing off with zilucoplan. The other advantage with zilucoplan is that you can use it with IVIG or Plex. So you can use combination treatment with zilucoplan. With the ravulizumab, IVIG can interfere with the mechanism of how the half-life of ravulizumab is longer through the FcRn receptor. So there's pros and cons with each of these treatments. Again, the cons are obvious. Meningococcal vaccines are necessary. Meningococcal B vaccine has changed its label now where you need a 6-monthly dose. So it takes 6 months to be really fully vaccinated. And the response may not be durable, meaning that when you stop treatment, the disease or the symptoms come back relatively quickly. So it doesn't sustain a long-term remission off treatment, but it's very effective while you're on treatment. The FcRn inhibitors decrease IgG levels, right? That's what they're supposed to do. So they can cause infections. So the rate of infections can be a little higher in patients treated with FcRn inhibitors because IgG levels are reduced. And because the treatments that are approved for FcRns are cyclical treatments. So with efgartigimod, you get one infusion once a week for 4 weeks. With rozanolixizumab, you get it for 6 weeks, but then you stop for 4 weeks. And some patients get worse during those 4 weeks. So there's reemergence of symptoms in some patients. In some patients, they don't even last those 4 weeks. And so that can be an issue in some patients with cyclical dosing. Some patients, some of them are here, but some patients can have a durable response. And so again, each patient, like I said, is a little bit different. It's tough to predict the responsiveness preemptively, but the cyclical dosing can be advantageous in some patients where they remain in remission for weeks or months, and they may need 2 or 3 cycles a year. But in some patients, there's reemergence of disease relatively quickly after each cycle, making it challenging. And then B-cell depleting therapies, rituximab is very effective in MuSK patients, not as universally effective in acetylcholine receptor antibody patients. And then enebulizumab, which may get approved next year, works a little bit higher up at the CD19 level. And it remains to be seen how we will use that in MG. But based on the RhinoMax study, one of the questions is whether using it early on in the disease process can change the tempo of the disease in the long term. So the question then is, when I'm seeing a patient, which is the best treatment for that patient? It depends on the comorbidities. If someone has diabetes, it changes what we do because steroids can worsen diabetes, hypertension. What is the disease characteristic? There are some patients who have severe worsening relatively quickly. I have patients who go into the ICU within a few weeks of initial diagnosis. Some patients have milder disease, which kind of lingers on for a while and then worsens. So again, it depends on, and then some patients have severe dysphagia and difficulty breathing. So it depends on the disease characteristics, depends on what treatments have been used previously. And then we take into account how intrusive the treatment is in terms of the patient's daily activities, occupation, and so on. So this is one strategy. I want to leave some time for questions. So this is one possible treatment strategy algorithm. One is, the first question in all patients is whether you should get a thymectomy. If you have a thymoma, you have to have a thymectomy. But if you don't have a thymoma, if you're young, thymectomy is a good option. The surgeons like the myasthenia to be well controlled before you can get a thymectomy. So sometimes it's challenging to get into a place where you're doing well and you can tolerate the surgery and so on. I always start treatment with corticosteroids or some prednisone, and I get an idea in 4 to 6 weeks, 8 weeks of how responsive the patient is to prednisone. If I'm lucky and the patients become near normal on prednisone, then I gently taper the dose of prednisone. And if I'm lucky and the dose can go down to 5 milligrams a day, that's that's great for the patients because they remain on a, pill once a day and with minimal side effects, and they're doing great. But that's not universal. A lot of patients need more than that. So the next step then is, if patients are not adequately treated or controlled with steroids, what do you do next? Do you do FcRn inhibitors? Do you do complement inhibitors? And there's pros and cons with both of these approaches. So depending on how severe the disease is, what the patient wants in terms of, treatment, every 8 weeks, 1 hour versus, cyclical treatment with possible reemergence of disease. With FcRns, there's a slight increase in risk of infections. With complement inhibitors, even though meningococcus is a problem, we are vaccinating for that. So the risk of other infections is minimal. So those are the pros and cons, and we kind of discuss with the patient and come up with a plan of which kind of treatment would be appropriate next. And then if the response to FcRn is not robust, then the next step is complement inhibitors. In some patients, we use complement inhibitors ahead of FcRn inhibitors. So it really depends on, it's tough to give you an universal algorithm, but I think each patient is different, and discussing pros and cons of these treatments and coming up with an individualized treatment plan is really the right thing to do. And then we talked about rituximab and B-cell depleting therapies as the early kind of treatment down the road. There's talk about doing meningococcal vaccines right at the onset of myasthenia so that it allows more flexibility down the road because it takes 6 months to be fully vaccinated now. And, the downside is that if you vaccinate someone and they don't need complement inhibitors down the road, then you're vaccinating, without any kind of benefit from the vaccine. But that's something to think about, whether we should use meningococcal vaccines in all patients with MG right at the beginning so that it allows us to freely use complement inhibitors down the road. So that's all the time I had, 45 minutes, but I'll love to take questions, comments, thoughts.
So everyone, we only have time for 2 Slido questions. So we will take those 2 Slido questions now.
Why do even the newer treatments often work for months, few years, but then stop, reduce working? Does MG change, permanent damage, hydrosis? It's tough to know what you're asking exactly, but I've seen that, say if someone is on, and I've seen this in a real patient where the patient was on some azathioprine and prednisone, and was treated with complement inhibitor and got better, got to normal. And the long-term immunosuppressant was stopped and the prednisone was tapered down relatively quickly. And the patient then got worse after that, suggesting that that initial response that was seen was not just from the complement inhibitor, but there was a contribution by these other drugs as well, which was not obvious when we saw when that patient was seen initially. So that is something to think about, whether you're doing something else, especially long-term immunosuppressants like mycophenolate. They take time to kick in, but they also take time to come, kind of go away from the system. So you have to be very gentle in tapering the long-term immunosuppressants. So I do it very slowly, like for mycophenolate, 500 milligrams a year, for example. So that could be playing into it. The other question in this context is whether the disease is worsening over time, which you can see, in patients with thymoma, if there's residual thymic tissue, which is now growing. But in general, in most patients, the most severe disease is in the first 2 to 3 years. In most patients, after the first 3 years, the disease tends to be less severe than the first 3 years in most patients. So I don't think the treatment stopped working, and I showed you those 6 cycles, and so it's unlikely that the treatment stops working and the disease is worsening. So it's tough to know as a blanket thing. Permanent damage. So if there is permanent damage to the muscle membrane, you shouldn't get a response initially either. right? So if you're treating someone with a treatment and they normalize, it implies that there's not a lot of permanent damage. So the permanent damage doesn't manifest later on after the initial response is positive. Higher doses, I mean, all of these newer treatments are fixed doses, really. So it's not like steroids where you're increasing the dose or IVIG and so on. CAR-T therapies could be a game changer. I mean, they, like I said, work upstream in the immune pathway. And so it could be a very good treatment for some patients, but they could be toxic. So I don't know how broadly they'll be applied. And I think the role for these other treatments will still remain. But I think in intractable patients or very severely affected patients, it could play a role, I think.
Thank you very much. Thank you so much for the presentation.
Rosenfeld, we sure would appreciate that. We have a real treat for you in this next talk. So it is absolutely my pleasure to introduce Dr. Jeffrey Rosenfeld. The MGFA works very closely with Dr. Rosenfeld. His talk is called The Changing Conversation, and I think you guys are going to enjoy this talk so very much. Dr. Rosenfeld has done talks at our National Patient Conference in years past. He's done a talk specifically on hope. For those of you who have heard his talk, Hope, you have come to us and you have personally asked to bring him back to do that talk again. It has been our highest ranked talk of all of our National Patient Conference sessions in the history of this program, which is amazing. And I understand why, because it is quite a moment when he does that talk. You just have goosebumps the entire time. It's really something special. We have that recorded. Those sessions are all on our website. If you haven't seen it, I encourage you to watch it. But today, Dr. Rosenfeld is going to talk to us about the changing conversation, and I'll let him explain to you what that is. Dr. Rosenfeld is a professor of neurology and the associate chairman of the Department of Neurology at Loma Linda University. So I'm going to invite Dr. Rosenfeld to come up to the stage. And just a friendly reminder that we do have Slido. You have to go back in at the beginning of each session to be able to upload questions.
Thank you very much, Sam, for that very kind introduction. I may stop here in my head, but and thank you all so very much for being here. You are my kind of audience. They say we're not taking a break and everybody gets up and leaves. That's my kind of audience. I really like that. I appreciate that. I could definitely have been part of this audience. So I am so very pleased to be here and be invited back again. And thank you so much to the MGFA to invite me to give a talk, which for me is a new talk. I've never presented this content before. So you certainly don't look like guinea pigs, but you are playing that role today as I roll this talk out. And I'm anxious to hear myself the feedback and the questions as to how it is received. This, as I say, is a new talk for me. And I just want to share a little brief story about this talk. When I'm in my career, whenever I'm presenting a new talk, a phenomenon happens that I'm not even surprised by anymore. I just sort of expect it. Usually something preceding while I'm working on the talk, deciding what I want to present, I have an experience that shapes or molds what I'm going to say. This was no exception. And I don't understand, I used to think it was coincidence. Now I think it's divine intervention, because about 2 weeks ago, I was thinking about what I wanted to present for this talk today. And I was giving a talk to our residents. I was not speaking about myasthenia. I was talking about a disease that's far less common than myasthenia. And I made a comment just by way of introduction to get into it. I said, this is a fascinating disease, the one I was teaching them about. And you don't want to miss it. And although you might not want to have it, you certainly want to recognize it when you see it, and you want to treat it because the treatments are effective. And then I was about to get into the content of what I wanted to share with them. And one of our junior residents interrupted me. She didn't raise her hand. She just interrupted me. She said in front of the whole room, "Excuse me, doctor, which one of our neurologic diseases would you like to have?" I didn't expect the question. And I thought for a minute, because I didn't think anybody was going to ask me a question in the introduction. And I thought for a minute and I said, well, you're right. None of the diseases that we treat you would elect to have. You wouldn't select the neurologic disease. But if you had to have one, and you're asking me that question, I think I might choose myasthenia. Why? Because there is nothing that we are experiencing in the neuromuscular space right now that is as exciting, as fast-paced, as rapidly changing as myasthenia gravis. And if I had to have a disease, although I didn't select it, I want a disease where a lot is going on. And that little interaction before that talk was what framed the perspective I want to bring to you today, which I call the changing conversation. Because the conversation has changed dramatically. We are, I think, by any account, living through an unprecedented time. And years to come by, when we look back on this time, we're going to say that was a Renaissance period, because I have never seen anything like as fast developing, as rapidly changing field as we're seeing in myasthenia. And I'll just leave you with one kernel before I get into what I really want to talk about today, the nature of the conversation. When we hear about all these drugs, and it can be a dizzying array of long words that are hard to pronounce, don't pay attention to the number of drugs you hear about. Pay attention to the mechanisms. Every time we introduce a new mechanism, it's not just a new bullet in the gun. It's a new gun. And we are introducing new mechanisms at a pace that I have not seen in any other disease at any other time in my career. This is an incredible time. Now, I'm going to take you through this change in conversation. Oh no, I thought this one was better. Sam, you broke it. You have to do a dance. We're going to hit on 5 points as we go through this little brief talk. I'm going to give you a little bit of a historical perspective on myasthenia, something you haven't seen today. And we're going to talk about autoimmunity and targets. You've seen multiple pictures. We're not going to spend a lot of time. I think you're ready to see why understanding autoimmunity has led to this incredible growth and excitement in the field. The concept of minimal symptom expression should be something that is emblazoned on your dialogue, emblazoned in your head as an important entity. This is today's optimal endpoint, minimal symptom expression, which is to say you essentially don't have symptoms. That should be the goal. And the conversation that I have with patients, especially new myasthenic patients, is I frequently start out to say, so that you know, my goal is that you have no symptoms. Doesn't mean you don't have the disease. It doesn't mean you don't have to take medicine to keep yourself symptom-free. But my goal is no symptoms. Now, I don't get there 100% of the time, but I get there a lot more commonly today than I used to get there, when in the past the conversation was, I'm going to make you better, life will be livable, and you might have to make some accommodations to your lifestyle. But now the conversation is, when I ask you how you're doing, I'm asking you relative to no symptoms. Not I'm doing a little better. I want you to say, for a period of time, I have no symptoms. And that's possible today. We're going to talk briefly about the difference, an interesting project that I'm involved in, looking at the difference in patient perspectives with myasthenia versus provider perspective. And then lastly, I'll end up with this, probably what you really want to talk about is, what can you do if you don't feel that your conversation is this conversation? So we're going to start by talking about, ooh, this is going to be a long talk. Maybe it's this one. Oh, okay. So we're going to talk about this timeline, which is very busy, and it's not really meant for you other than to realize the slide is very crowded. And the slide is very crowded because a lot has happened in myasthenia. And where I'm circled there is the first description of what we today believe is myasthenia, and it was in the 17th century. Because even earlier than that, there was a description of an Indian chief in Jamestown that had the disease that today we probably would call myasthenia. But in 1672, there was a clinical description. That was a long time ago. And then, as you heard this morning, Dr. Walker identified physostigmine, which is similar to pyridostigmine, mestinon, in 1934, a long time ago, but still one of the mainstay of early treatment as a milestone. And then in 1960, it was the autoimmune part of myasthenia became in vogue, and we knew that we could, or it was discovered that there's an antibody that is affecting the neuromuscular junction, and then shortly thereafter, we could transfer the disease using that antibody, injecting it into a rabbit. The rabbit would get sick as well. So the concept of a pathogenic antibody that was causing a disease, an autoimmune disease, has been around for a while, but nothing like what's happened recently. This slide ends off at 2019. We're going to take it a little bit further than that, but there were many, many discoveries that happened fairly rapid fire after that. There was the discovery of different types of acetylcholine receptors. There was MuSK antibodies that were discovered. The FcRn was the system was first introduced in the early 2000s. And on the bottom, you can see the medications and how they evolved, with the earliest one being in 1954 when physostigmine or pyridostigmine was introduced, and then cyclophosphamide, azathioprine, IVIG, rituximab, all of your usual friends that you're familiar with, you can see when they were born. But the pace was fairly good. And I remember as a trainee in neuromuscular, I was thinking, you know, of all of the diseases we treat, myasthenia, we really understand quite a bit about it because of this. We understand why it occurs. We understand how it occurs. We understand how to treat it. That was way ahead of where we were with other diseases. So the short story is we were doing pretty well before the recent past that I'm about to explore. Now, I'm very grateful to my friend and colleague, Dr. Ali Habib, who's at University of California in Irvine. And Dr. Habib gave a talk recently that I was attending, and he had some of these slides are his because I was so impressed by the way he synthesized the information that I asked him if I could use his slides. So very grateful for him. We're going to see several of them. But what this slide shows is the number of treatment trials classified by the mechanisms over time. And the short message is, if you look at what happened in the past year or two, there are more clinical trials, more investigations just in the past year or two than there have been essentially in the 10 or more years before that. And before that, we thought we were doing well. But what happened in the last year or two with drugs, as you see there, both complement inhibitors, FcRn inhibitors, new mechanisms that we'll talk about in a moment, it has been an almost exponential rise in the discovery, in the pace of discovery, and in the introduction of new mechanisms. The names in those boxes are either the name of the drug or the name of the trial where that drug was studied. The important thing is what's happened in the most recent past. Now, this is a cartoon similar to what you've seen before. I'm sorry, my pointer doesn't work, so let's take you through. We talked briefly earlier about the importance of the thymus in terms of the immune system, but generally speaking, in broad strokes, the immune system is broken into two components, T cells and B cells. Easiest to think about, T cells oftentimes stimulate B cells. They'll find something that's foreign and they will present it to the B cell, and that B cell will then go on and form antibodies to that entity that the T cell has found. It's kind of like the infantry on the ground found the foreigner and they brought it back to headquarters and they're going to mount the response. Now, B cells form antibodies in plasma cells. They're a type of B cell. And those antibodies, in the case of myasthenia and other autoimmune diseases, are pathogenic. That means they cause the problem. And we've been speaking about that all morning. That problem is an attack on the neuromuscular junction. But what I like about this cartoon is it shows where the different medications act, where we think the best mechanism of these medications act. You can either suppress the T cells so that they don't stimulate the B cells. You can deplete the B cells you could affect the antibodies that are the result of the B cells. In the case of pyridostigmine, you can actually let the acetylcholine last longer in the neuromuscular junction. And then complement is a downstream. Complement is like the final death knell for the neuromuscular junction membrane. After the attack has occurred, complement comes in and wipes out the path. It's like a major bombing attack. So we have drugs that are listed here at all of these stages. And it gives you some appreciation of where you're affecting what part of the process. Now, this is an unnecessarily busy slide, which is really just to tell you in broad strokes, there are three main foci of therapy. And they're listed here in the Roman numerals. You can actually affect the way the antibodies are produced or not produced. That is, affect the way that either by affecting the T cell or the B cell. You could activate or inhibit complement, which would minimize the downside of the antibody. So you're suppressing the antibody's pathogenicity with inhibiting complement. Or you can promote the exodus of the antibody with recycling. And we have, again, different medications. And when new medications are coming on the board, usually they fall into one of these three categories. And on the left, you can see some of the more recent advances in treatment-wise. Those are the names of various trials that I'll spare you the detail of, except to say they represent a number of different mechanisms that affect at least one of these big picture areas where you want to inhibit this autoimmune inflammatory response. So let's look at what we have and where we're going. We know that this FcRn, you've been hearing a lot about FcRn this morning, because FcRn, when it was introduced, the FcRn inhibitors, they were game-changing in that we never had this mechanism before. This wasn't just, when efgartigimod was introduced, it wasn't just a new medicine. It was a new way to treat the disease. We didn't have another medicine that did this. Subsequently, rozanoliximab is another FcRn inhibitor, and we didn't have any before that. They can be and have been, in my hands, quite effective at patients that I wasn't able to bring under control as well. That would be enough. But in addition, we have two that are on deck. Two drugs that I expect we're going to hear about shortly within the next year or so, as to whether we now might potentially have as many as 4 different FcRn inhibitors. Complement inhibitors you've heard about, and as mentioned in my earlier slides, complement is kind of a downstream event that basically takes out, it's very destructive to the cell and the membrane. Complement is a very powerful immune mechanism, and we now have 3 medications that are approved that affect the complement system. They're not identical, they're not the same medicine, but they all rely on the same mechanism. I'm going to talk a little bit about what differences in complement drugs are in just a second, but that would be enough, but we have 4 more on deck. Now, one of them, the bottom one, I think that trial ended. I don't think that was a successful trial, but we have 4 other drug trials that we're going to hear about in the next year or so, affecting complement differently, but taking what was good in terms of complement inhibition and better efficacy and making it even better. Now, as I mentioned, complement, I don't really intend to understand this, for you to understand this, except that understand complement is like a cascade. So there are different components or events that occur in complement, and they go from, they're numbered C1 through C9, I think. Maybe there's no C10, I don't think. There's C9. And it's a cascade. So some people feel that if you block the cascade at C5, you have the best benefit. Some drugs may block the cascade upstream at C1, and you'll never get to C5. But it's a way of inhibiting this very pathogenic mechanism. So when you hear about new complement drugs, and you are definitely hearing about new complement drugs, you only need to walk through the exhibit hall and you'll hear about several. You're hearing oftentimes about drugs that affect the cascade at different levels. And we're trying to figure out which is best. What a good problem to have. Now, there's one drug that I really am watching for, the so-called NIMBLE trial, because the NIMBLE trial is a trial of a combination therapy. Now, for reasons I don't have time to elaborate on, I'm a real fan of combination therapy. I like combination therapy because in a disorder like myasthenia, but others as well, likely more than one pathogenic process is happening at the same time. There could be complement destroying the membrane. There could be new antibodies being produced. There could be T cells activating. So having a drug that does more than one thing, which is what this drug is, it's the combination of two, one affecting the RNA and one affecting complement together. We're curious to see how this works out. So this is another one estimated to be completing at the end of next year. It's a complicated design, and I'll spare you it, but I think we're going to get an answer at the end of this. Now, B cell, we heard a little bit about just a moment ago about Rituxan. Rituxan has been studied in the past, and what Rituxan does is it reduces the production of the antibody. It suppresses the B cell activation. We also have another medication in the MINT trial, which is just, I think, recently completed or completing and is available now. And there's one on deck. So where we had good news before, we have better news now in every single mechanism. Now, COUR has a very interesting approach, and forgive me if there are representatives of COUR Pharmaceutical here. I'm not going to do this justice, but the approach that COUR is taking is called immune tolerance. Now, immune tolerance means that we are going to make your immune system not care about the acetylcholine receptor anymore. We're not going to suppress the Bs or the Ts or the complement. We are going to turn down the volume a little bit on the immune system by immune tolerance. If this is successful, yet another new mechanism to bring to bear on this disease. Another new mechanism in a disease where we already have an abundance of therapeutic mechanisms. This is a very interesting trial because of its novelty. And similarly, as has come up before, there is a variety of cell-based therapies. Now, cell-based therapies, CAR-T, which was asked in a previous question and was referenced before, that is yet another new mechanism for treating the disease. And what CAR-T therapy is we are going to take your cells and we're going to program them to basically stop the immune attack on the acetylcholine receptor. So your own cells are curing your own autoimmune disease because they're reprogrammed cells. This is a therapy that's used in cancer. CAR-T therapy is used in cancer therapeutics because they're programming the T cells to attack the tumor, shrinking the tumor down, and it's been very effective. It's a newer idea in autoimmune disease, but we don't just have one attempt at programming cells. You can see the number and the variety of therapeutic initiatives in this area. Now, I want to change gears and get back to the conversation because I told you a little bit about the pipeline. And as I was preparing and thinking about this, I came across this really interesting paper for two reasons. The paper is looking at patient perceptions. And the way it looked at patient perceptions was not the typical way, which is to ask the patient what they think. What these folks did is they went to social media, the thing we tell you not to do, and they went to social media and said, what are people saying who are writing about their disease on social media? So they went to lots of different websites. It's interesting, different chat groups and all the places that you're going to, even though we asked you not to do that. And they looked at over 13,000 conversations about myasthenia. And they said, what are common in these conversations? Now, mind you, they did this recently. So they had a lot of reasons to think I would have thought, that people are going to be like, what's this? What's this? What's this combination? You know, what's going on here? We hear about all of these exciting developments. Now, admittedly, patients that write into these things probably, if they really are positive, they're probably not as inclined to write in as if they're really negative. So there might be a little bias. But what they found was negative sentiments about the disease, about the treatment, about the current state of affairs. Negative conversations predominated about 60% of the time. Positive thoughts about what's going on in the field, as captured by this method, they used an AI tool to analyze. I don't think they read 13,000 messages. I think they used the AI tool. Positive conversations were somewhere less than 10%. I think I had it in there, 2%, I think it was, or yeah, 2% were positive. 2% of the conversations were positive. 60% of the conversations were negative, accepting the bias that I mentioned. And most people did not write about what was happening in the field today. Most people wrote about problems they were having with their myasthenia. Now, if there is not another reason for a wake-up call for a new conversation, when I read this, I thought, wow, what's going on in the world is not what I see in the clinic. Now, here's a little bit more of a breakdown from that paper. You could see in the different categories what they wrote about. Most people wrote about issues of their symptoms and issues of the impact on their life. Very few people, except interestingly, steroid-treated people and IVIG people, wrote about their treatment. Now, probably the steroid people wrote about their treatment and were negative because they were getting negative side effects from the steroids. But where's the box that said, oh my gosh, what are all these new drugs? They didn't capture it. It wasn't positive. Now, this is a score sheet that you probably are very familiar with. This is a picture of the MGADL score. This is our simple 8-question questionnaire that probably many of you have answered when your clinician or nurse asks you these questions. And basically, it's a way for us to keep score. It's a way for us to keep score about how you're doing in total. It doesn't capture everything that happens to you. It's just a snapshot of where you are. And it's a way on the phone in my clinic, the nurse can tell me what the score was and what it is now, and I get a sense of how fast things are changing. It's not perfect, but it's pretty good. And it's used as the benchmark for most of the clinical trials that we have been doing and that we are currently doing. But the important thing is the notion of minimal symptom expression, which has become very much a topic in the most recent past, ever since the efgartigimod trials. Minimal symptom expression is when you have a 0 or a 1 on the scale. Now that means 0 means you are normal and have no symptoms, and a 1 means that you have maybe an intermittent problem that doesn't really get in your way and might be mildly annoying, and we lump that in with no symptoms. But the concept of minimal symptom expression is something that has become the new standard, in my opinion, and as well it should become the new standard, because it means that you have this disease, but it is in control. Now, these are some images from the efgartigimod trial, the ADAPT trial, and I think you, Dr. Muley, showed some of these before. There's something I want to point out to you without even getting into the difference. Each of these graphs represents a different one of the outcome measures in that trial. And what you want to notice is the blue line dips down quickly. The takeaway message is this drug acts quickly. Within the first cycle of treatment, these people got dramatically improved in multiple endpoints. In fact, on this slide, every endpoint illustrated, they improved dramatically. And the magnitude of that improvement, you can see by how many points they improved. This is a plot, Christmas tree plot, where you can see the 15%, to lose the top on the right there, 15% of the population improved by 9 points on the ADL is a huge improvement. So this was not subtle. This got a lot of people's attention. And this is why this and other data from this trial was really highlighted the importance and the magnitude of the impact of FcRn inhibition. But the thing to tell you is 40% of the responders in that first cycle, 40% of the responders had minimal symptom expression. I've been practicing for just over 30 years. I know I look younger, but over 30 years, I have never seen this before. I haven't seen it in any disease before, let alone myasthenia, which is a chronic ongoing disease because of the immune system. Very powerful. Now, if we look at the rosanoliximab study, similar result. A quarter of the patients achieved minimal symptom expression, improving from a score initially of 8. Now, I didn't do this justice because subsequently, if you keep taking it, perhaps that number is bigger than a quarter, with subsequent dosing. But very dramatic. These really move the needle. And here again, in the same study, we see that there's dramatic and fairly precipitous improvements early on in the conversation. Now, think to yourself, if the magnitude of these improvements in patients that might never have seen this kind of improvement with other therapies, if that isn't the reason to change your discussion about myasthenia, what is? This is very striking. Now, I want to digress for a moment and tell you just briefly about a project that I'm involved with. And I have to be a little careful because the results of this project are just coming out now. They're not published, but I thought it was so relevant to a discussion about a changing conversation that I wanted to share it with you. And this was a project where we surveyed 200 patients and about, I think, 100, and the next slide I'll show, 150 or so healthcare providers as patients. 140 providers, 200 patients. And one of the questions that was asked in this survey was, if the ADL score of 10, which is pretty high, it goes up to 24. It's 8 questions. You can get a 3 as the highest. So it goes up to 24. An ADL score of 10, would that be acceptable to you? 19% of the patients said yes. 80% of the patients said no, that is not acceptable. But the surprise was 31% of the providers said, yeah, that's okay. That means 31% of the providers thought that's as good as it could get. An ADL score of 10 or more. And honestly, I get that because for a long time, some patients, we couldn't bring them back towards normal. So for a long time, this would have made sense to me. And I might have answered, if that's as good as it gets, then that's as good as it gets, because I was counseling patients to, we're going to improve your life. And you might make some accommodations, and we would talk about energy conservation, and we would talk about all these different interventions with perhaps other therapies, but we didn't move the needle as much as we could now. So this was very recent data that we collected. The other thing, which was an interesting question, was what do you think the likelihood of your reaching a minimal symptom expression is? So 30% of the patients said, yeah, that's possible. And only 50% of the providers said that's possible. So as I'm thinking about what is the changing conversation, this is the changing conversation. How is it that there is such a disconnect between what's going on and what people are thinking, if not that the conversation is not what it should be? There are a lot of treatment options today for myasthenia. There are almost as many on deck coming. There's a lot of reason to be optimistic, aggressive, hopeful, some combination of those things. So we've talked about the historical perspective of myasthenia, always an interesting disease, always with lots of interval stages of milestone progression. We talked about the accelerated advances in the recent past. I mentioned about minimal symptom expression because that is the new standard, or should be the new standard. Not everybody reaches a minimal symptom expression, but everybody should want it and should strive for it. You'll get closer that way. I mentioned briefly this disconnect between the patient perspective and the physician perspective, and there's more to come on that story because we're just still analyzing the results of some of that feedback. Now, what can you do? Probably this is what you want to know. Supposing you're sitting there and you say, well, I've never had that conversation in my clinic visits. Set the bar higher. You know, I mean, you're already a motivated patient group. You came to this meeting. That already puts you ahead of what the average patient does. Is the room would have to be much bigger if this was everybody. So you are a motivated patient group. You are the ones, among the ones, who should be going to the clinic and saying, why can't I get to minimal symptom expression? Although my colleagues may not appreciate this, you should challenge them. You should say, what about some of the newer biologics? What do you think? Is this appropriate for me? Can we try it? Maybe there is a reason why they're not doing that. You want to first set the bar higher for yourself, then set the bar higher for your provider. And I have an expression in my clinic that we use frequently, not just in myasthenia, and I say frequently, if you get your head in the right place, your body follows you. So your head in the right place here is to say, I want to do better if you don't feel you're doing as well. And I know some of you may have been dealing with symptoms for years and years. And just ask yourself, could it get better? Should it get better? How could it get better? That's what you're challenging your provider to do. Expect minimal symptom expression. Use it in your discussions. It's not to say that everybody will reach that. But again, everybody should want to reach that. That is your expectation. And persist. There are a lot of possibilities. I tried to do a flyby to show you all the different things that are on the pipeline and all the tools that we have now. There's more to come. There's no indication that there's a waning interest in this disease. So while, as I started to say, what neurologic disease would you want? Nobody wants a neurologic disease. It's like going to the dentist. Nobody wants to see the neurologist, right? But if you had to have a neurologic disease, you want one with a heightened interest, with a rapid pace of discovery, with a rich pipeline, and a wonderful support network, which is what you have here within the MGFA. So, insofar as you might wish you weren't in this room to begin with, or the need to be in this room to begin with, you're in the right place. You're in the right place, and I would say at the right time. So I'll leave it there, and I very much look forward to your questions and hope this was helpful. Thank you very much. Any questions? Oh, we're going to look there. Is there a correlation or data on the impact of COVID and COVID vaccines to the increase of MG cases? I'm not aware of any formal correlation. It kind of begs a question that's beyond what you asked, but the question is, when you have one challenge to your immune system, can another immune system mediated disease get worse? Which is sometimes why if you have an infection, for example, like a pneumonia or an upper respiratory urinary tract infection, we see an uptick in the way to control the myasthenia. It's because your immune system has got some lighter fluid on it now because it's fighting multiple things, and so it's all revved up. Not specific to COVID, and I personally have not seen any convincing data, or any data for that matter, on the COVID vaccine with respect to myasthenia. In seeking MSC, how do we avoid making perfect the enemy of good? When should we settle with effective but imperfect therapy? Well, that's a hard question to answer from the podium, but I will tell you, what is the harm in wanting more than you have? Well, if you're consumed by it, yeah, that's a harm. But what's wrong with going and challenging yourself and your provider to say, could it get better? So I don't really think there's a time when I want to say to my patient, I think we're done. And there isn't a time. I'll try something else. I'll try a different combination. There's another trial that's ending. Maybe we'll try that. So I don't know if there is an issue with wanting more than you can have, any more than there is an issue with if you had another chronic illness, supposing you had diabetes, for example. You have diabetes and your sugar is good. It's not great, but it's good. Do you say we should stop trying to get the sugar great? No, it's a chronic issue. But the important thing is your head is in the game. Your head is saying, I want this to be better. And the likelihood of achieving it, I think, is greater. Can damaged muscle or cell tissue be restored? Yes. So you heard from Dr. O'Connor earlier about how the junction actually gets degraded by this complement attack. If the immune system is turned down, if the immune system stops attacking the junction, it can rebuild. It can. It's not permanent. Did I answer those? Oh, there's one on top. Is there research into long-lived plasma cells when to escalate beyond IgG targeted therapies to agents like CD38 inhibitors in severe MG with epitope spreading? I don't know. Probably that makes sense. Dr. Connor's in the back, but do you have an answer to that? There's a microphone back there.
Can you read it again? I can't see it from back here.
It says, is there research into long-lived plasma
Right.
cells and when to escalate beyond IgG targeted therapies to agents like CD38 inhibitors in severe myasthenia with epitope spreading?
Okay, so Anonymous is a scientist. That's a good question. CD38, that gave it away, right? So long-lived plasma cells are a type of B cell that we've been talking about today that make antibodies. And the concept is that they're very hard to target therapeutically. And so you have to come with new ideas. So we've been talking about, you've heard about CD20 and CD19 today, and these are real, they're happening. But CD38 hasn't been targeted directly. And I think the question is whether or not there is research looking at other subtypes of B cells that are very important in producing the antibodies that cause the disease. And the answer is yes. And I reiterate what's happening here today about how this is a great time for the research moving forward so quickly. So yes, this is being investigated.
Thank you. I was going to say that. Any other questions?
Thank you. Thank you so much for your time. Thank you very much for the presentation.